Hi, it's Mrs. Vance again. We're back to Unit 8, the flow of genetic information, and this is the second part of notes, which is about the process of transcription. So think back about what we said in the previous lesson, okay, that we've got to make a copy of the DNA. The DNA is too large to leave the nucleus, so we have to copy the part of DNA into RNA in order to make the protein. Okay, so we're going to transcribe an RNA molecule from a DNA template. And this is similar to, it looks a lot like the replication of the DNA. Okay, there is an enzyme that will take care of this called Rna Polymerase. Rna Polymerase actually does several things. It basically unzips the DNA molecule. It puts in place the complementary RNA bases, and then it zips the DNA molecule back together and releases the RNA. And so this allows us to make a copy that we need to. Now, there are specific sequences along the DNA mark that signal where transcription starts and stops. So it is more complex than we're going into here. There are a few more things involved, but you don't need to know about that as long as we can understand the process of transcription. So here's RNA polymerase. We're going to represent it by this kind of egg-shaped molecule here. Basically, it's going to unzip the DNA here, and it's going to plug in complementary bases, and then it's going to zip the DNA back together and release our RNA just to be used to make the protein. It's transcribing in this direction, basically, so it's unzipping here, transcribing, and then zipping it back together over on the right. Okay, we'll watch a video, first of all, that shows you how this works. It's much easier to see it sometimes if you see some kind of representation of it while that you can think about while you're learning about it.
What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So that kind of shows how the whole process works. The blue molecule that it showed there, of course, was RNA polymerase, and the yellow molecule that was coming out was the messenger RNA. So messenger RNA is one of the 3 kinds of RNA that we talk about in the cell. We abbreviate it with, like here, with the lowercase m with RNA. And basically, it codes the amino acid sequence, and it also takes the message from the DNA to the ribosome, which is the translation machine of the cell. Now, in prokaryotes, like bacteria, it happens in the same place that messenger RNA is made. But in eukaryotes, the messenger RNA has to get out of the nucleus first via the nuclear pores. Now, one thing that's different about eukaryotic messenger RNA is that it has some intervening sequences or interrupting sequences called introns that separate the coding regions, which are called exons. And so we have to edit the messenger RNA in eukaryotes before it leaves the nucleus to make sure that we have only the parts that are coding for the protein before it attaches to the ribosome. So the processing is called RNA splicing, and basically it removes the introns, those intervening sequences, and joins the exons together. And this produces a continuous coding sequence. It's not necessary in prokaryotes because they don't have introns in their DNA. Then we also, in RNA splicing, there's also a cap and a tail of extranucleotides that are added to the ends of the messenger RNA that allow it to signal the nuclear pore that it needs to go out, and also protects the messenger RNA from enzymes and helps the RNA bind to the ribosomes. Big thing is there are enzymes in the cytoplasm that are programmed to hunt down and chop up stray pieces of RNA. That's part of your cell's defense mechanism against viral infections. So here's basically what happens, okay? There's our original DNA up here. Our exons are the parts that are going to be expressed. That's the expressed part of the gene. That's the coding region. The introns are the sequences that need to be removed. And so basically we're going to add this cap and tail of extranucleotides there that help protect the messenger RNA from the enzymes in the cytoplasm. And then there are enzymes called spliceosomes that actually pull out the introns and then splice the exons back together. Once that happens, then it's ready to leave the nucleus and go out and be transcribed. And we have one more quick video that shows basically how the splicing process works.
Once a gene has been located and transcribed into mRNA, it must first be edited before it can be translated into a protein. This editing process is called splicing. It involves removing non-coding regions called introns, leaving only the protein-coding exons. In the cell, the introns are removed by special enzymes which recognize specific sequences. These enzymes cut and rejoin the coding regions for translation into protein.